It's my pleasure to present a phase 1/2A trial and update on HESC-derived RPE in advanced dry AMD patients with geographic atrophy. We'll go through those acronyms as we move forward. So this is on behalf of all the employees within BioTime, both within the United States and Israel, and all the participating investigators and the clinical trial staff. Our safe harbor statement can be found at biotime.com, by the way. Above all, we'd like to thank all the patients who are participating in these studies. So dry AMD, age-related macular degeneration, is a progressive disease that ultimately will lead to blindness, and is the leading cause of blindness in the developed world at this point. It goes through various stages as you progress from a primary cellular event to secondary cellular events, which can then lead to functional impact. And as you go, the RPE cells, are the retinal pigment epithelial cells, are the cells first impacted by the disease, then photoreceptors are damaged, and then you get an area of geographic atrophy thereafter. We're studying the administration of a suspension of highly purified derived RPE cells, which are grown in animal-free conditions and are administered by a subretinal injection. This is an example of the progressive nature of geographic atrophy. This is a subject just over the course of 4 years. This area will continue to expand until ultimately they will lose all vision. I'd like to thank some of the preceding speakers for covering most of this, but the bottom line is that the RPEs support the photoreceptors. As they, through environment, through genetics or whatever, begin to lose their ability to metabolize byproducts and keep the photoreceptors healthy, they begin to degrade, and then that ultimately results in the areas of geographic atrophy. So what's the goal? So the goal of is try to support the dysfunctional and degenerated RPE cells. The drusen are these fat lipid areas that form when RPE cells get sick. And those are hallmarks of the disease, and we'll show you some signs and symptoms of that on our OCT imaging. The goal when RPE are dysfunctional is to ideally replace them with healthy young RPE cells, supporting the photoreceptors that are still there and allowing them to continue to survive. Again, we've used a directed differentiation process, has been previously published, to generate the RPE. They are extremely pure and have undergone rigorous sterility testing and karyotyping to make sure that they haven't drifted at all over time. The agency we've worked with closely to go through a variety of studies to prove that when give the cells, they are totally purely differentiated RPE cells. We've also conducted a number of studies in preclinical animal models. The Royal College of Surgeons rat model with genetic variant that is as close to AMD as you can get. We've been able to demonstrate in a time-limited fashion, because of the genetic deficiency of the disease, that we can show both a structural and a functional benefit in the rats. Part of the hallmark of this is the ability to phagocytize the rat rhodopsin. And so this is a rat model, but again, human RPE cells that have been administered and are actually phagocytizing the rhodopsin. As part of the safety studies as well, we looked at the RCS rat, the skid mouse, and also pigs before we began to give to humans. The areas stained in red are the RPE cells that were administered. The current design of the phase 1/2 study is an FDA, again, approved study. It's being conducted in Israel and the United States. It's a concentration finding, safety, and efficacy study of RPE cells, again, given subretinally to subjects with advanced dry AMD and GA, via vitrectomy and subretinal delivery. Again, the primary endpoint is safety, and we're looking at additional efficacy endpoints as well. Inclusion, exclusion, as you might expect, advanced dry AMD with GA. Safety and tolerability is the key primary endpoint, but we also are looking at preliminary efficacy studies as well, the area of GA over time, best visual acuity, and also a variety of other parameters such as functional reading tests, microperimetry, and studying the ellipsoid zone. So this is a sample example from first cohort. The top images are the preoperative images, color fundus, and they show below the intraoperative creation of the subretinal bleb where we administer the cells subretinally. This is an OCT image following at baseline, 1 day post-op, 2 weeks, and 1 year post-op, showing that where the retinopathy occurred, that it healed relatively rapidly within usually 1 to 2 days. So what do we see? Part of the yellow dotted line, I wish I had a pointer, sorry, shows the area where the subretinal bleb extended to, and then follows through up to 24-month time point, 2 years. And you can see there are areas of hyperpigmented and hyperechoic areas that are suggestive of RPE cells, and I'll show you why here in the next couple slides. So this is the first one is baseline, that one's 2 years later. You can see that the dark pigmented areas around the areas of GA, we think, based on some of the data I'll show you from the pig, are the RPE cells that have been settled in. The areas indicated in purple are the areas of geographic atrophy. You can see that the cells suggestive of the implanted RPE cells are indicated by the blue arrows. The green arrow is indicative of an epiretinal membrane, which is one of the adverse events we've noted in our study, but that hasn't resulted in any surgical or medical interventions to date. What's key to this, too, is, again, you've got the 2 year time point in the lower right there, and we've got evidence of cells 2 years out without any immunosuppressive therapies after the initial administration. Again, you can see the pigmented cells visible there on the top part where the bleb was created. So one of the reasons we think that they are functional RPE cells is based on our data from the pig model. So on the upper left is the surgical bleb created in the pig eye. You can see the demarcation of where the bleb was extended to. OCT imaging, the yellow arrows are indicative of where the cells were deposited. And the image on the bottom lower right is that from one of our first human subjects. The thing about the pig is post-mortem we were able to sacrifice the animal and perform immunohistochemical analyses and able to demonstrate the RPE cells that were present. This is a subject from the third cohort. The areas indicated by the yellow arrows are the drusen, which again is what's the hallmark of the disease as it progresses. You see the baseline image, the 1 month image, the third month image, and then month 4.5. And what you see is a restoration to more normal architecture. And so we're following that over time. So when you look at it in real time here, the top is baseline, the lower image is month 4.5. As the green line goes up, that's the area of OCT image going throughout the bleb. So it's not just a cherry-picked area, it's the area throughout the entire treated area. So we'll go to the next slide here. So why do we think that it might be a potential result of the therapy administered? On the top, you have the lower macular scans. So those are the areas that did not receive RPE cells, the OpRegen cells. On the lower bottom are the area where the bleb was administered, and we think that RPE cells were functional and deposited. On the left side, you see the baseline, and on the right, the month 4.5. Again, it's a restoration of more normal architecture. What this means on a functional basis, we're trying to determine. As far as systemic and ocular safety events, thus far, no treatment-related systemic adverse events to date. Unrelated SAEs, as you might expect in an elderly population. No unexpected ocular adverse events to date. Expected adverse events that have occurred are related to the epiretinal membranes, which I described. Some were new occurrences, some were slight thickening. Again, none have interfered with their distorted vision. The early cohorts were legally blind, so it's hard to disrupt vision there. But as we've moved to better vision cohorts, it's obviously an area that we're closely following. There was a pigmented epithelial detachment in one individual, but they've occurred in their untreated eye as well. So again, one of the hallmarks to follow the safety is, did we have an impact on vision over time? We always treat the eye with the poorer vision, and basically it has remained stable over time. So the summary of the phase 1-2 observations to date, we're currently in cohort 4, is that the OCT images show that the administration of the subretinal bleb is rapidly healed within 1 to 2 days, essentially. fundus findings, different imaging modalities would suggest that there's a variation as the eye begins to heal following the bleb administration, then that stabilizes over time, and that's demonstrable out to 36 months. The BCVA, so it's best corrected visual acuity, and the area of GA have remained stable in both the treated and fellow eyes, or progressed at the rate that you would expect. The subretinal pigmentation, it correlates with an irregular subretinal hyperreflective aspect of the OCT, is evident in the treated area, and it has remained stable for greater than 2 years in some subjects. And why we think this matters is because from the animal data that we've seen, the histology, it would suggest that it is indeed RPE cells. So we have seen new or worsening ERMs that have not required surgical intervention at this point in 7 of 9 patients. And the subretinal transplantation appears to be well tolerated to date. There's potential early signs of structural changes, which again will need to be correlated with a functional benefit over time. So with that, I'll take any questions.